It's a real pleasure to be here. You good? Okay. So when people ask me what I do, I say, well, I'm a clinical geneticist, which means that I work at Texas Children's Hospital, and I see kids with all kinds of different genetic disorders. But I also have other roles. So I teach in medical school, and I run a research lab. In a research lab, we use mice models and humans to try to understand things. And you say, well, what do you exactly work on? And I would say, you know, if you really want to know what I am, I'm a gene hunter. And people laugh and they say, what's a gene hunter? I said, I spend most of my time trying to figure out what genes are important for human health, and when you hurt those genes, what happens? Like, what happens to patients when they're missing that gene? And so, of course, that fits in really well with 1p36, because 1p36 deletion syndrome is all about missing genes. And so, in this particular case, I want to be talking about how we hunt for genes in this modern age. And I really like these first two pictures. Both these guys are actually really good hunters. They're just in different environments and they use different tools, right? But the guy on one side is no really better than the guy on the other side. It's just that they understand their environment and they use their tools right to be able to accomplish what they're doing, which is hunting. And so in the same way, we use the tools that we have available in the best way possible to try to find new genes for 1p36. So how do we do this? Well, so on one side is kind of the old school way of doing things. And some of you, your children were diagnosed in the old school way. I'm always shocked when I come to these conferences, and we must be all getting older, because all the 1p36 deletion kids, they're all getting older on us. I'm like, wow. Like we have 30-year-olds walking around here. I'm like, how old am I then, right? Because we've been doing this forever. And so some of you actually your kids were diagnosed this way. And so some of you know a chromosome analysis. That might have been the way that your child was diagnosed with 1p36. It could be that they were diagnosed by FISH, but there's limitations to that. Most of the kids who are diagnosed this way have terminal deletions. In other words, they have deletions that take on the very top tip of chromosome 1, or they're very, very large. So there's limitations to this. And it's also very difficult to say how long or how big those deletions are, at least doing it very accurately. So a chromosome analysis, for example, what does that look like? These are chromosome 1s. And some people say, well, you know, my child has a really big deletion. You should be able to see that. These are actually very well prepared chromosomes. So the way you look at chromosomes under a microscope, some of them can be really stretched out and have very great detail. Others are super scrunched and you can't see very much. But 1p36 is this little area right here. And the change that we're seeing is something that goes from here to this. I don't know if you saw that, but it's like literally, it's like that little tiny piece, that's it. Like that little tiny piece disappears. And so some people are diagnosed that way, and it takes an expert who looks at these and says, yes, that one little piece has disappeared. That's a chromosome analysis. FISH, and the way you write that out is you'd say this is 46 chromosomes, this is a male XY, has a deletion on chromosome 1 that takes off that tip. FISH is a little bit different. So some of your kids were diagnosed by FISH. The way this works is you actually use these little fluorescent probes, and they light up both sides of chromosome 1 in this case. You can see that one of them has these pink ones. That's the correct side for the bottom end, but only one has the green side, and that's because the green is this 1p36 deletion. So the chromosome that has both is normal, and the chromosome that does not is abnormal. That's that little piece right there. Again, FISH just lights up one piece of the chromosome, and so you can't tell how big it is. You just know that that one piece is missing. So this is the way we do it in the old days. How do we do it nowadays? If your child's probably less than 10 years old, let's say, it's much more, or maybe even 15, it's much more likely that you had a chromosome microarray that was done. And many of you are very familiar with this. These can do both terminal deletions, which is the tip, but also interstitial deletions, which come kind of in the middle of chromosome 1p36. It can tell you both the location of the deletion and how big it is. So these are very, very powerful. How do we do a microarray? So most of your kids will have done this on blood samples, but recently we're doing this more on saliva samples. For a blood sample, we're interested in getting the white blood cells because they have the DNA. And then we extract the DNA from those white blood cells. And then we use this laser scanner. This laser scanner costs more than my house, like probably 2 times as much as my house. Not everybody has one of these laying around, but these labs have laser scanners. And then we use this little tiny glass slide to do the microarray. This glass slide has thousands of probes on it, sometimes up to 1 million probes, and it kind of works like this. This is a 1 million probe microarray. This is a picture of one. And if you actually blow it up, you can actually see there's little tiny circles. Those circles are printed by a giant inkjet printer. And they actually build up the DNA one base or one letter at a time, all the way up to about 60. And each one of these circles represents a unique part of the DNA. You can see that there's different colors, right? So some are red, some are yellow, and some are kind of green. So if I just kind of eliminate everything else, the green means that there's 1 copy of the DNA there. Now you're supposed to have 2, one from mom, one from dad, so this would be a deletion. 2 is normal, and 3 means, or red, means that you actually have 1 more copy than you're supposed to have. So kids with Down syndrome, for example, they have 3 copies of chromosome 21. So they would be a bunch of red on their microarrays. Now you can't look at these spots and know anything about what's going on. So you got to have a computer do this for you. So this is what it looks like when we sit down at a computer and look at these arrays. And the dots over on this side, each one of those dots is 1 point of the DNA. So if you blow this up, the black dots in this case are normal. The green dots are a deletion. And these red dots here and there probably don't mean much because they're just kind of hanging out. They're just kind of spurious things. But you can see this child has a deletion, and you can actually mark it off and say that's exactly where the deletion was or is. And then you can actually look at the genes that are inside that deletion. This is how we do a microarray. I used to do these by hand, like sitting on the floor in my, I would do these just because they were by research. And my kids would come sit by me, and they're like, hey, look, there's a deletion. I'm like, yeah, you're right. And when they got to college, their professors would show these. And they're like, so does anybody know what this means? And my children, there's more than one, would raise their hand, and he's like, what do you think this means? He's like, well, it looks like it's a deletion on chromosome 2, probably in band like 3-6. And they're like, no way. Like, how do you know that? And they're like, oh, my dad's a geneticist, and we used to do this with him, like hang out, read arrays. It's like, this is pretty easy. This is not rocket science. Like, you can do this, I can do this, my kids can do this, right? Okay, so what do you get when you get the report back? I often ask families to send me the report. What am I looking for? I'm looking especially for this box. This box tells me exactly where the deletion begins and ends. Sometimes people are a little bit confused because microarrays cannot test the very tip of the chromosome. You have to have, each chromosome is capped by these repeat DNA segments. And so sometimes the deletion is a little bit bigger than what families expect because they can't put probes in the very first part of that DNA. So most times they'll say things like, it's deleted, and they'll give the first marker on chromosome 1. So in this particular case, what I look for is I look for these minimum intervals and maximum intervals. I don't know if you can see this, but I kind of blew it up. And so here you can see there's minimum interval and a maximum interval. This really helps me. I can see the size, I can see where it begins and ends. In this particular case, this is an interstitial deletion. But anyway, it's very, very helpful for us as geneticists because we know exactly what we're dealing with when we see one of these. So if I ever ask you for that, I'm looking for that box. That's the most important part. So chromosome analysis microarray analysis is a good way to go. So what do we do as far as like questions? The biggest question that I get asked at these conferences, and almost always there's someone asked this, why doesn't my child with 1p36 look like anyone else? Now, some of you say, well, hey, everybody looks exactly the same. And the answer is yes. But every once in a while, there's a child who's just dramatically different. And they're like, my child doesn't look like anybody else. And my answer is, it's because they don't have the same syndrome. And they're like, what? And I was like, yeah. As it turns out, almost no one in this room has the same syndrome. And you're like, well, why are we all at this conference? And the answer is because it's all involved with 1p36. But every single one of our kids are unique. Why? This is what chromosome 1p36 looks like when you blow it up. It spans 30 megabases, which is 30 million base pairs, 30 million letters. Some kids have a deletion that may take in 99 genes. That's a small deletion. Other kids have much bigger deletions that may take in 186 genes. Those are the terminal deletions. Some kids have interstitial deletions where they take in, let's say, 77 genes, but they're in the middle of the chromosome. Each one of those is considered 1p36 by a doctor, and even a geneticist can say that. But we all know that those are three very different deletions, and they can act very differently. There are some overlaps and such, but this is a very key thing. This is why when I visit with you, I'm like, I want to see your microarray result, because what genes you have deleted may make a difference in what I do for your child. And literally, there is not a single 1p36 deletion that's the same. They're all unique. So this is why if you want your child to be different, you can be different. They can be completely different. So let's talk a little bit more about this. No two children with 1p36 are the same because no two 1p36 deletions are the same. What I want to know is which genes are responsible for the problems we're facing. In other words, are all 99 genes important? The quick answer is no. There's some genes, when you delete them, they don't make that much difference. And there's other genes, when you delete them, they make a big difference. I want to know the difference between those, and I want to identify those particular genes so that when a child comes with me or a parent comes and say, my child has this little baby deletion, does that mean something? I need to be able to say yes or no, and it's going to depend on what I know about the genes in that small deletion. So, why do we care about genes? I'm going to just kind of say it the same way. If you know what a gene does, you can tell what problems a child might encounter if they had that gene deleted. That might and if are really important because not always when you delete a gene does it give you the exact same problems. So some of you say, well, my child has seizures. And you say, I want to know the gene that causes that. And the problem is there's probably more than one gene that causes that, but even kids that have that gene deleted may not get seizures. Why? Because there's all kinds of other genes that are working in every single person, all 20,000 of them, and sometimes genes do a little bit better or do a little bit worse, just depending on all those other genes and what they're doing. Environment, diet, you name it. Lots of things can make a difference. So might and if are great words. So could this change something? Yeah. It could change the initial tests we run. It could change the surveillance plan, which means like how we follow kids. It could change the therapies we use. It can change a lot of things. And I'm going to give you some examples of exactly how that works with different genes. So how do we used to hunt for genes? I've actually done almost every way to hunt for a gene because I'm old. So I've done old school ways and I've done new school ways. So let's just talk about some of the ways we do this. One of the ways we do this is the old school, is we find super large families that have the same problem Sometimes these families are consanguineous, meaning that they actually are inbred. These large families are very powerful genetically. We do what's called linkage, which tells us what part of the DNA is affected, and then we look at candidate genes and we screen them. This is old school. I've done this, but I'll just tell you, I do not know of a single 1p36 gene that's been identified this way. This is good for some things, but as you can imagine, most of you are not from families where you have large numbers of 1p36 kids, right? This is just something that we don't use that often here. Instead, we use this second thing, and this has been used a lot. This is where you take deletions from different kids and you line them up. And you say, well, all these kids have some special phenotype or problem. Let's say it's seizures. And you try to figure out what is always in common with all those kids and their deletions. This is very powerful. And so we're looking for what's called a common interval, or a piece of DNA that's always deleted. Then we say what candidate genes are in that piece of DNA, and sometimes we can go right from the deletions to the gene. Let me give you an example. So cardiomyopathy is something that we often talk about for kids that have 1p36. This can be a very dangerous problem. It can sneak up on kids. And if I remember correctly, we had one child who had a free echocardiogram at one of the 1p36 conferences and was taken to the hospital because he was having problems with cardiomyopathy. Did not know it, but was actually in quite bad condition. Cardiomyopathy means a problem with your heart muscle. Not a hole in the heart, but the actual muscle's not working right. So cardiomyopathy is a problem. A really smart group back in 2013 said, we're interested in cardiomyopathy. We're going to take all the kids who have had deletions and we're going to line them up. And we're going to hope to find the gene that causes this. So they did just that. All these are 1p36 deletions. They lined them all up, and lo and behold, there's like this one little area that keeps on lining up. They said, what's the genes that are in that area? One of the genes was a gene called PRDM16. In fact, it was the only gene that was in that area in every one of these children. And they're like, that probably can't happen by just coincidence. So what they then did is they said, well, this PRDM16 gene looks like it's pretty important. Let's go ahead and we're going to take this common interval and we're going to do another study called a cohort. So a cohort, you pull a bunch of people that have that same problem and you pick a gene that you think causes it and you screen all those people. So in the same study, they went and they pulled a cohort. In this case, they pulled a cohort with cardiomyopathy, 75 individuals, and they said, how many of them have changes or mutations in PRDM16? This is the PRDM16 gene, and every one of these black dots represents a person that they found that had a change in that gene. And so by doing two different techniques, they proved it two different ways that PRDM16 is important for cardiomyopathy. So why do we now care about PRDM16? If you're going to listen to one particular part of the talk, this may be the most important one because it's the one that we make the most common decisions about. A child might develop cardiomyopathy if their PRDM16 gene is deleted. That means that we need to obtain an initial echocardiogram, and as far as we know, that child needs to be followed every 3 years with an echocardiogram, just to make sure that everything is going okay and we don't miss something. So that's typically what we do. And that means that you're going back for an echocardiogram quite frequently. It's unlikely, though, for a child to develop cardiomyopathy if their PRDM16 gene is not deleted. In that case, we just obtain one initial echocardiogram. If there's no structural, like holes or anything like that, and they're cleared by a cardiologist, we don't have to do echocardiograms anymore. And so one of the things I do when I get kids that come see me is I'm looking, is that PRDM16 gene anywhere near their deletion? If it's in there, we're going to be visiting a cardiologist and we're going to have follow-up. If it's not in there, if it's far away, I can pretty much say this is not going to be a problem for you. And that's, again, a lot less visits to the cardiologist. We pass that information on to the cardiologist and they'll then make that decision. But usually these kids will be cleared and you don't have to go do this again. So these are the old school ways that we used to do things. And we still use these things now, but they're old school. How do we do the new stuff? So finding genes these days, we also use individual patients. So I can see a single patient in clinic and begin this process. We then use what's called exome sequencing to identify the problems in our patients. In exome sequencing, we're actually checking all 20,000 genes in a single test to say, do they have changes in that gene? It's pretty powerful. We've had this test for, I'm thinking about 7 years maybe. When it first came out, it was considered experimental, but now we use this every day in the hospital. And so when a child comes in with something that I don't know, I don't recognize that pattern, this is one of the tests that I might ask for. Exome sequencing. We then use databases, really large databases, to help us to identify the genes that are important. This is not rocket science. You can do this just like I do. I teach medical students, I teach undergrads, I teach high school students to do this. Like, you can do this. Even my kids can do this, right? I'm going to teach you how to do this. You don't have to if you don't want to, but if you want to play, I'll teach you how to do it, okay? Here we go. The one thing that's also required for this is networking, because one patient will not be enough. So we'll talk to you about networking too. So how do we do modern gene hunting? The basic idea is that there's a bunch of people out there somewhere that have changes in a gene. They could be anywhere in the whole world. But if we were to pull them all together and then start to look for problems, we would find that they have similar problems amongst them. That's the idea. I'm going to only see perhaps one of those people, but if I can see one of them and I can connect with everybody else, we could solve this together. That's the idea. The initial thought about a gene might come from a mouse model that we make. I work with mice in our lab. It could come from a laboratory finding, or it can come from a human patient just like I was talking about. I'm going to give you examples of all three. So let's start with the mouse. So how do we work with mice? Once upon a time, and this is a true story, once upon a time there was a mouse who was blind. This mouse was made at Baylor College of Medicine and had 2 changes in a gene called RERE. It causes microophthalmia, which is small eyes. It's mapped onto chromosome 4 in mice, and the specific change is in a gene called RERE. This gene is super important because it turns on other genes and turns them off according to what they need for development. It turns off and on eye genes so that eyes develop correctly. It's important in the vitamin A pathway, and that pathway affects a lot of different organs. So this was an interesting mouse for us. As it turns out, in humans, mice and human chromosomes are different. On mice, it's on chromosome 4. In humans, it's on chromosome 1p36. So it sits in 1p36, and we're like, hey, we know this syndrome. And specifically, it fits in what's called the proximal critical region. This proximal critical region is basically a region where if you delete it, you get all the symptoms of chromosome 1p36 deletion, but the kids look a little different. And so they don't have the same look as a child that has a terminal deletion. We knew about this, and we're like, hey, RERE sits right in the middle of this. What kind of problems do kids with these proximal deletions run into? They're the same ones with terminals. Eye vision problems, poor growth, cognitive impairments, brain anomalies, yada, 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 yada. Well, we already knew that the eye and vision problems could be traced to this gene because of the mouse, right? But we had the mouse in our lab. So we're like, why don't we check all the other things and see how many things this gene can do? because maybe it's responsible for a lot of problems that we see in kids with 1p36 deletions. So we started to do that. I'm going to focus just on one thing, and I'll show you how we did this in our lab. Let's start with just cognitive problems, which almost every child with 1p36 has got some. Brain anomalies sometimes, like on an MRI you might see something, and then these delays in motor development. What could we learn from our mouse? So first we looked at mouse brains. I don't know if you look at mouse brains, but we do. So we took a look at these mouse brains. They were actually the brains before the mouse is even born. So these are embryonic brains. And if you can see the top ones, that's the ones that are normal, and the ones on the bottom are the mutant ones. You may say, Dr. Scott, those look like kind of the same thing. But when you do the measurements, they're actually smaller than they're supposed to be. The mice are the exact same size, but the mouse's brain is actually smaller at every one of these stages. And that's what's shown on the graph on the other side, where we look at the weight of the brain and the size of the brain, they're smaller. And we're like, that's interesting. We then said, well, is the brain formed correctly? There's a part of your brain kind of back here called the cerebellum. It's important for your fine movements. If any of you know anybody who has Parkinson's, this is the kind of fine movements that this part of the brain directs. It's called your cerebellum. It's supposed to have these little divisions. When we looked at our mice, they didn't have all the divisions they were supposed to have, and their whole cerebellum was smaller than it was supposed to be. And so the little star shows some of the folds that's supposed to be there, and our mice just don't have those folds. So this part of the brain is not formed correctly in RERE mice. Then we said, well, what about the neurons themselves? There's these neurons called Purkinje cells. They're really big. You can see them, and you can stain them. And so they're great to look at. On the top are wild type neurons. They have all these branches. They look like trees. On the bottom, you can see that our mice had smaller branches. And sadly, the person who worked on this project, he says, Dr. Scott, to tell you the truth, the way we usually do this is that we count every branch. And we see if the number of branches are different. I'm like, you're kidding. He goes, no, this is how they do it. I'm like, so you're going to count every branch? And he goes, I guess so. I'm like, you are a very dedicated postdoc. You make that happen, and you come back and tell me. And so he literally went, and with a special thing, he just counted every single branch off of every one of those neurons. And when you do that, yeah, they actually have less branches too. Each one of those branches is a connection that helps these mice to help coordinate their movements. These are abnormal nerves. RERE is important not only for brain development, but how the nerves get set up and how they function. So, for our mouse, we knew that now it has vision problems, and they also are cognitively not normal. Their brains are abnormal, and they also have some delays probably in their development. But it matches with the things we're seeing. We then checked all the other things. In this mouse, every single one of them is found in this mouse. Problems with growth, problems with cleft palate, problems with hearing loss, problems with cardiovascular problems, problems with cardiomyopathy, renal anomalies, it had it all. And we're like, dude, this is the 1p36 mouse. This is it. It can do it all, right? And so we're like, yeah. So we said, everything is present in our mice. And then we said, hey, we want to get more money to study this mouse. And the people who reviewed our grants said, well, it does it in a mouse, but does it do it in humans? And we're like, well, duh. And the answer is, prove it. The problem was, nobody screens this gene. This gene is huge. And which one of these problems are you going to make, how will you gather the kids that have this? It's so rare. How will you know which ones they are? And so we were stuck. And I said, I can't move forward. I can't do this because there's no way we'll ever identify these kids. Until exome sequencing came by. Remember I told you that we screen all 20,000 genes in one test? As soon as we started to use exome sequencing, I said, it's just a matter of time. I just have to sit by the phone, maybe for a couple years, and someday it's going to ring, and someone's going to say, I have a patient that has a change in RERE. And so I just waited, like for a long time. And finally, the phone rang. It actually didn't ring in my office. It rang in the office of a woman down the way. She was a genetic counselor. She got a call from another genetic counselor, and they were talking about a specific gene. At the end of the conversation, the genetic counselor says, hey, we just recently saw a kid that has a change in a gene called RERE. Do you have any kids that have changes in that gene? The genetic counselor who works with me says, no, but I know the guy who has the mouse. Is like, would you like to talk to him? And she's like, sure. So she comes down to my office and says, hey, someone just called and they have a patient that has a mutation in RERE. I'm like, my phone just rang. Oh yeah, like I've been waiting for 2 years for my phone to ring, right? And today's the day. So I called the doctor up. His name was Elliot Scher. He's a neurologist. And I got on the phone with him and I said, let me guess. Your patient has eye vision problems with his growth, cognitive impairments, probably has a brain anomaly, yada, yada, yada. And he goes, not everything, but I've got a lot on your list. I said, I've got the mouse, you got the patient, we should work together. So we did. Now, one mouse and one patient is not enough. So we began to work together to say, there's got to be other patients that are being seen. So these are the first patients that we're seeing. All of these kids were identified by networking, and all of them had these kind of problems. -2, we never found one that had cardiomyopathy, and we never found one that had renal anomalies. Now we have even more kids, and the renal anomalies, yes. The cardiomyopathy, no. It looks like PRDM16 is still the only gene that we know on chromosome 1p36 that causes cardiomyopathy. Interesting. But anyway, these kids helped us understand that, yeah, all these things are possible with just deleting one gene. So what does that mean? Well, first of all, we should say, so we published this, and this little tiny like red line that you probably can't see, it says, if you delete RERE, you can have problems like hypotonia, seizures, behavior problem, structural brain anomalies, ophthalmological anomalies, congenital heart defects, and genital urinary anomalies. Kids have any of that? The answer is yeah. This is the gene, or this is one of the genes that's a critical gene for 1p36. In fact, when you line up the problem list, it's like almost identical, right? So this is a super important gene on 1p36. That's where it sits. If your child's deleted for this, this is important. So why do we care about RERE? So like, how does this help us make decisions? A child may have many different medical problems if their RERE gene is deleted. So how does that change management? Definitely need an echocardiogram. They need hearing and vision screens. They need to have scoliosis checks on a regular basis because scoliosis is a problem for RERE. We see that especially in our teenagers. They need to be monitored for seizures, and of course, pretty much every child needs to have early childhood intervention, right? But this is kind of our list. If you're missing RERE, we need to be thinking about these things. Most of these are just typical stuff we do. But again, if there's something missing on this list, I need to check that off because this is what this gene does. So that's a mouse. How do we go from like a laboratory result? So I'm going to start with that same mouse. So this mouse is this RERE mouse. I don't think you're probably familiar with looking at hearts, but the top heart is a very happy heart. It has two sides and it has this big middle piece that runs down the middle that divides the right ventricle from the left ventricle. The bottom one where the black arrow is, do you see how that connects both sides of the heart? That's against the rules. That's a ventricular septal defect. So our abnormal RERE mice have this. So we said, well, if RERE is important in heart development, and we like to do heart development in our lab, What are the genes that are downstream that is turning on or off? So we took embryonic hearts, checked what genes were being turned on and off, and we found that they had lower levels of certain genes that were being turned on in their developing hearts. One of the genes was a really cool gene called SPEN. And we're like, SPEN's very, very low. And so we took a look at SPEN mice, and the wild type mice have normal hearts, but the SPEN mice have ventricular septal defects. So it looks like RERE turns on SPEN. If you move either of those, you end up with heart problems. You say, well, Dr. Scott, who cares? Well, SPEN's on chromosome 1p36. Like, wow, hey, we work on that. So it's like, yeah, this is pretty handy, right? So we got SPEN mice in our lab, and we were working with SPEN mice, but we also started collecting kids that had problems with SPEN. It was a gene that was not known to cause any defects in humans when we started. So what do? We want to know, is SPEN a gene that may play an important role in 1p36 deletions? And specifically, we need to answer this question. Does losing one copy of SPEN cause medical problems? Now, that's a really good question. Because again, remember, there's lots of genes that are missing in kids with 1p36, right? We want to know if losing just one copy is enough to cause problems. How do we do that? How do you identify bad 1p36 genes? Now, this is a little complex, but if you focus for a little while, you can do this. Not that hard. Ready? There's a database out there that has 141,000 plus patients or individuals who are, I'm going to use my air quotes, normal. If you said, Dr. Scott, are you a normal person? Most people would say no. Like, I do way too many crazy things. I've had 5 surgeries in my life. But for this study, I would be considered normal. Why? Because I'm living my life. I have a family. I have a job, yada, yada, right? So most of the normal people in this group have medical problems, but not critical medical problems. They're just kind of normal people, living their life, doing stuff. But that's helpful. Why? Because they did exome sequencing on these individuals. And the thought was this: if they have mutations in genes that cause major medical problems, they won't be in this normal group. They'll have been removed from the normal group because they'll say, oh, you don't qualify because you have a massive heart defect, or you don't qualify because your eyes are not developed correctly. That's the idea, and that was the power of this. Every gene has a certain size, and because mutations happen randomly, some of us are going to have changes in that gene that make it not function. And by the size of the gene, you can actually predict how often that's going to happen. So you can take things and say, well, I have a gene, and based on the size of the gene, we predict that 45 people out of this group will have a change that will make that gene not work. Then you could say, well, how many people do we actually see in this group? If it's pretty much the same, well, then that gene doesn't do anything really critical, right? It didn't take that person out of the normal group. Instead, they're still hanging out, having their families, going to work every day. Like, this is not a bad gene. So we would say this is not bad gene, and then we give it a score. The score is called a PLI score. Just all you need to know is it ranges from 0 to 1, 0 good, 1 bad. And so this would be a 0. I'm going to look at this gene. If your child has this gene deleted, I'm going to say probably doesn't make a big difference. In contrast, if we look at that same group, and there's only 5 people in that group that have a change in that gene, that means, in other words, 40 people were removed from the normal group because they had a change in that gene. That means that gene probably does something important. So in this case, this is considered a bad gene, and it's going to get a PLI score of 1 or really high, and it catches my attention. So that's how I look at things. So anytime I see a kid who has a deletion, if there's only a few genes in it, I go and I say, is it a bad one or is it a good one? Can we lose one copy without causing a problem? How do we actually do that? Well, let's just take a few examples first. Remember, PRDM16 causes cardiomyopathy. Good gene or bad gene? Bad gene. Should have 49 people, only have 4 in that group. That's bad gene. And we would expect that, right? Because cardiomyopathy would move you out of the normal population because you're having a problem with your heart. They're going to say, no, you don't qualify. How about RERE? Bad gene. 65 because it's big. 3 people have a change in that gene. It's like, that's a bad gene. PLI score 1. So what about SPEN? This is the actual screenshot from the database that we use. And so we go in here. Anybody can do this. You can do this at home. It's called the Nomad browser. And we pull up this little part right here, and it says there's supposed to be 129 people with changes in this gene because it's a super big gene. We only find 4. That's a big difference. That means 125 people were removed from the normal population in this study because of a change in this gene. So that means this is bad. It gets a PLI score of 1, and it means I'm ready to have another phone call, right? Because this is a bad gene. Now, this time we don't, well, modern day, we don't have to wait for the phone to ring anymore. Instead, what we do is we actually get proactive, and we go onto a different site called Gene Matcher. This was genius. I did not think this up. I wish I did, because it would be really cool to say, I thought of that, but I didn't. The idea is really simple. You put your email in, you make a password, and you put in any gene you're interested in, and it matches you with everybody else in the whole world who has interest in that gene. So it's like super cool. So I would put SPEN in here, and I say, hey, who are all the people that are interested? It sends me a list of all their emails and names. I then email all those people and say, hey, I got a patient that has a SPEN change. Has anybody else got a patient with a SPEN change? And if they do, we start working together. How did this work? These are the first 34 individuals who have damaging variants in SPEN. Well, these are the pictures of the ones that will let us use their pictures, right? But we gathered 34 people in a very fast way over the course of, let's say, a year and a half or 2 years. And all these individuals have changes in SPEN. Pretty cool. So what do? We write a paper. This paper defines all the changes that happen with SPEN. Let's see. The clinical profile of this disease, blah, 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 includes developmental delay, intellectual disability, autism spectrum disorder, anxiety, aggressive behavior, attention deficit disorder, hypotonia, brain and spine anomalies, congenital heart defects, high narrow palate, facial dysmorphisms, which is just the way your face looks, and what I think is probably most important and interesting, obesity and increased body mass index, especially in females. We were sitting around yesterday and we're like, how many obese males do we know who have 1p36 deletions? You may help us, but we couldn't think of any right off the top of our heads. And it's like, it's interesting. SPEN has a really unique function. You probably know that females have 2 X chromosomes, right? In order to make that work, you have to inactivate one of those chromosomes so you're balanced. This plays a role in inactivating X chromosomes. And when it does not work, females are actually different and have different problems than the males do. One of the differences is this obesity problem. And so it would be really interesting, and it's not something that I've done, but to see if that actually makes a difference, and if SPEN, like having this deleted or not deleted, makes a difference in whether there's an increased rate for obesity or not, especially among females. But amongst the patients that just have SPEN mutations, that's the case. So, what does this mean? Why do we care? You're going to see a pattern forming here. A child may have many different medical problems if their SPEN gene is deleted. Management would include echocardiogram, scoliosis checks again, ADHD medications might be a helpful thing, nutrition consult if there's problems with obesity, and of course, early childhood intervention, which is pretty much, I think everybody should have that probably, right? Okay. So that's how you go from a mouse, but specifically a laboratory result in a mouse, to finding a brand new 1p36 gene. Last one, just a patient. Single patient walks into clinic, right? So single patient walks into my clinic. Actually, he doesn't walk into my clinic. He's 16 months old, and he should be walking, but he's not. He also has no words. 16 months, you should have some words. We usually say kids should be walking by 15 months, and if they're over that, it's considered delayed. So he comes to see me. I'm like, yeah, this does not look right. We do an exome sequencing test, and we get a change in a gene called SKI. It's like, boy, you guys really come up with great names. We have 20,000 genes to name, right? And so, yeah, we got SPENs and REREs and SKIs and whatever. So SKI gene. This particular one, that bunch of letters basically says he has a change in his gene that causes part of the protein that this makes to be made, and then it stops. When the body does that, it often just says, this is not right, and it actually won't even make the protein at all. So this is a bad change. This change is similar to what would be if you actually deleted the SKI gene. So where's SKI? It's way up at the top. Most of our kids that are going to have terminal deletions are going to have SKI affected, as compared to SPEN, which is way, way, way down at the bottom, right? So does it have a bad PLI score? The answer is it's 1. It's a bad gene. So what do? Go to Gene Matcher, man. So I literally went in, we put this thing on Gene Matcher, and I said, hey, I got a kid who has a change in SKI. So far we have 17 individuals who have what's called loss of function variance in well, that's supposed to be SKI. I try to catch all my errors, but I don't do a very good job. Sorry, that's supposed to be SKI, not SPEN. But these patients have not yet been published yet. So if you go right now onto Google, you won't find any abnormality associated with SKI loss of function. But we have the 17 individuals. What do they have? They have developmental delay, intellectual disability, autism spectrum disorder, ADHD, obesity, seizures, heart defects. This is probably, again, another gene that's going to be important for, well, all those different things. Hasn't been described yet. Working on it. Probably going to happen within the next year. So why do we care, or why will we care about SKI? Again, a child may have many different medical problems if their SKI gene is deleted. What would we do? We're thinking echocardiogram, ADHD medication, nutritional consult, monitor for seizures, early childhood intervention. You say, Dr. Scott, all your genes do the same thing. I can't help that. It's the way it is. It's the way it is. Which is probably good, because that's why we can actually come here to these conferences, and even though our kids do have different deletions, there's a lot of stuff that we're facing that's the same, right? But it's different genes that may be actually causing these things. And if we were ever to have like special ways of correcting this, we'd have to understand the genes that were involved in order to be able to do so. Okay, so take-home messages. We're learning more and more about important 1p36 genes. We have a lot to learn. I have a list of about, I would think probably 30 genes that I follow for 1p36 that are in various stages of being described. And so I keep tabs on these, and when we get enough patients together, we publish them. That takes a lot of work, but this is what we do. I'm a gene hunter. It can start from a mouse, of finding a single patient. It's driven by this clinical genetic testing that we do nowadays. This exome sequencing is a really powerful thing that we do. And networking is vital. If you looked, I don't know if you noticed, but all those papers that we published, most of them have 60 to 70 authors. Why? Because we're networking with 30, 40, 50 different groups from all over the world. This is typical. One of my grants asked me, is there any possibility that you might publish with someone outside the United States? And if so, give their names. And I'm like, I can't. There was a time when they asked us, list all the people you've ever collaborated with, give us a list. Mine was like 200 people long. And I'm like, and I still don't think I did it all, right? Because nobody does this alone anymore. We're all just working together, just like all you guys come together, right? We all come together as scientists to try to figure this out. So with that, I would be happy to take questions. We did pretty good. We started 15 minutes late and we only ended 6 minutes after the hour. That's good, right? But if you have questions, I'd be happy to answer them. You can also catch me. I'll be here for at least until dinner-ish time.
If you have a question, I want to bring the microphone to you.
So my son was diagnosed by FISH testing. He's almost 20. If I went back to a geneticist and said, could you run so that I can see specifically what's missing, would they do that? Or would they say, you know what it is, we're not going to do that now?
So yeah, the answer is yes, you can do that. Would it be helpful? The farther along you get on your age, the more you know about what's going on. There still could be some things that could be important. For example, that PRDM16 gene, if he's not being followed by a cardiologist, it might be an important thing to find out.
He has everything on that. That was really interesting.
And
I was just curious.
so it kind of depends. What's the cost of that? The cost out of pocket is somewhere between probably about $750, I would think I could get an array, probably something like that. Your insurance would probably pay for it. The older you get, the harder it is to get insurance to pay for these genetic tests. That's the bad part. But yes, could it be done? It could. And could it be useful? It could give us some information that might be helpful. Yeah.
Ken.
Behind you.
Sorry. That was my question.
Oh, okay.
Just a quick follow-up. Our daughter was tested with a microarray. Is it worth getting an exome sequencing?
No.
Okay.
So you have a different, so the only time when we would go to exome sequencing, okay, so there's two different things. There's actually two different tests that we run on any child who I don't know what they have. So if I walk into clinic, this is what I teach my residents. If you walk into clinic and you look at the child, you take the history, and you have no clue what they have, you're going to order two tests. One is a microarray, which looks for big changes, big deletions or duplications. If you find it on that, you may stop. If that doesn't work, then you're going to move on to exome sequencing, which then goes much smaller and looks at all the genes and does it one by one, and we're going to look for small changes. The only time that we usually trigger like an exome sequencing on a child that has a known deletion that causes disease is if they have something else that just does not have anything to do with that problem. So for example, if your child had, trying to think of a really good example, I don't know, something really weird with their gut. I don't know what I would even pick, right? It has to be something that's not 1p36. Then I say there could be what's called a dual diagnosis. In other words, this is a child who doesn't just have one problem, they've got two. How often does that happen? On kids that we can't diagnose who actually go to exome sequencing, about 6 to 10% of them have two diagnoses, not just one. That's why the geneticist walked into the room and said, dude, I don't know this pattern. Why? Because it was two patterns mixed. That dual diagnosis is something we think about, but a lot of people trigger these tests because they just haven't done their homework to say that's actually part of the disease you already know about. And so on a regular basis in the hospital, I get people to say, I'm freaked out because of this one thing. I asked my residents, I said, go to Google, put those two things in together and tell me if it's possible. And they'll come up and say, there's 3 papers that show that that can be done in this disease. And we write back and say, there's no point in playing. Like, we already know that this causes this. It's just, we learn. Over time, you learn what to expect. But if it's something really wild, you might, for most of us, we would never trigger exome sequencing. Yeah.
Are you open to collaborating with other geneticists that may not be as well versed in 1p36?
Most of the people I collaborate with have no clue what we're doing. Why? Because they are pediatricians who are overwhelmed. They see way more kids than I see every day, and they have a kid who has a change in this one gene. And they're like, oh my goodness, and I say, would you help us? And they're like, do what? I said, we just need you to help us make contact with your family. And they're like, sure, if that's all I need to do, I can do that. And so, yeah, we actually have all kinds of different levels. There's some people who play with me all the time, and we do lots of different things. And I'm like, yeah, I know you. But most of them are pediatricians who are just good people who want to do good things for their families. So, yes.
Awesome, thank you. And then one other, the database that you recommended, is anyone able to access that, or is it only doctors?
Yeah, so the databases that I've talked about, the one that you do the PLI score of 0 or 1, that's a public database. Gene Matcher, I think you have to record who you are.
Okay.
I don't remember if you have to be a researcher or a doctor.
Okay.
Yeah, so the question was, do you have to have some kind of medical background? You might have to for that. All the people who come up on those searches for me are mostly physicians and scientists, and that's helpful because I can then narrow down very quickly exactly who I'm looking for. So, I ask you for a patient. Oh, if you are a patient.
Okay.
Okay, there's lots of different sites that are similar to this. I like Gene Matcher the best. There's another couple that are coming out that might actually be more powerful. At the bottom of Gene Matcher, it lets you check 6 boxes that say, do you want to search all these other databases? I'm like, sure. And then I get emails. Every day. Why? Because I follow about 110 to 120 genes at any given time. And so there's almost not a day that goes by that I'm not talking to somebody in the world about some gene. And when we get enough patients together, we trigger it and we say, okay, we're going to focus and we're going to publish this gene. And as it turns out, a lot of times those are 1p36 genes because that's what we like to do. Yeah.
Our daughter Paisley is followed at University of Iowa, and she sees the cardiologist, ENT, every doctor possible. Is that because her geneticist or genetic counselor recognized some of the genes in her microarray, or are they just hitting all the hot button items?
So first of all, I like the University of Iowa because that's where I was trained. So that's always nice to hang out with Iowans, right? Okay, so most of our kids are going to see multiple specialists, right? Some of those are because we actually noticed things even before they were tested. And so there's certain things that they're going to have to be, I don't do neurology, for example. If your child has seizures, you do not want to come to me for their medication because I just don't know it. So it's a little bit of both. Sometimes it's triggered by exactly what we know is going on. And we'll have a kind of a list that we need to go down for every gene. So again, if you have one of these, you should see a cardiologist, you should get an echocardiogram.
Right.
Or you should see an endocrinologist if you have growth problems because that's the right thing to do. Stuff like that.
And we see all of those and many more.
Yes.
And I just didn't know if they were just hitting everything or if there's somebody's actually looking at her microarray and laying those out to build that checklist.
It's probably a little bit of both.
Okay.
So they probably saw the 1p36, they'll go through the list. I may go through the list a bit more in detail, but the one thing that really makes a difference is that PRDM16 gene. That's the first thing I check because it helps me and I make decisions right away off that. But yeah, it's not unusual to have lots of specialists. We like that. And when they're done and they say, we don't need you anymore, they should sign off and say, you're good. That's what you hope for. Yeah. Yes.
So a little bit in line with others, how often do you see your patients?
How often do I see patients?
So you see them once, right?
And
then they come up for follow-ups or not?
So typically what I do is I see patients based on how good I think their parents are. And what do I mean by that? There's some parents who are very with it. I can see them once, I can set them up with their specialists, and they will follow through and be amazing. For them, to see them once or twice, once every year is fine because I love you and you can come see me, right? But I don't worry about you. And so after they've done this for a little while and they kind of get this going, I say, hey, if you want to come see me because you just like me and I like you, we can do this. But I recognize that your life is busy, right? And you're already seeing this guy and this guy and this guy and this guy. Although I love you, I would be okay with you seeing me every 2 or 3 years. You have a pediatrician. If he or she ever thinks that there's a question, you can come back and see me immediately, or we can call. So I would say typically once a year, but once things are going, every 2 to 3 years would be fine. But I've got to have you be a good parent. If you're a bad parent, I'm going to see you more often because I don't trust that you're going to do what you need to do for your kid. And there are some parents, not 1P36 parents so much, but there are some parents that, dude, I just don't trust them. And so I see them much more often. Yeah, because I want to make sure their kids are okay. Would you think that's about right? Would you agree? Okay, and I am definitely not I'm not a typical pediatrician. So that you should talk about the different models. In our university, I work at Texas Children's Hospital. It's huge, and we typically don't see kids very often. If I go to Guatemala, which I'm going to do on a medical mission trip, that geneticist sees kids every 3 months. And I'm like, why? And because he says these families need help and support. I'm going to see them every 3 months, and they love him so much they will literally come at 6 or 7 o'clock in the morning. They will wait for him, and if he sees them at 4 o'clock in the afternoon, I'm freaking out as the American, right? I'm like, they've been waiting for how long? They're like, we love you, Dr. Silva. Thank you so much for seeing our child. We love spending the entire day waiting for you. And I'm like, wow. And like, this is not an American model, right? But they love him, and he does great things. So I'm going to hang out with him this next week, and we're going to see patients together. Yeah.
And one more quick, is there a very good source for clinical trials?
Oh, is there a very good source for clinical trials?
Yes.
It's just science. It was science.gov? Clinicaltrials.gov. Clinicaltrials.gov is the only one I know. And so anytime a patient asks, I do that. There may be other trials that are going on that are known inside here, though, that are not necessarily clinical trials, but are research studies and such.
Probably last question.
And I can hang out. You can ask me questions. I dressed as uniquely as I could today so that I can be picked out. So the guy in the white kind of interesting shirt, that's me. Yeah.
Yeah, doctor. Probably a follow-on to the clinical trials question. I was curious about gene therapies, and it's probably a follow-on to that one, right? The clinical trials?
So gene therapies are becoming more and more common. What are the challenges? So most of the gene therapies right now are targeted to one gene, and they have to be targeted well. Problems. You have to get, if it's a gene therapy, you have to get a change in a gene in the right tissue at a high enough level to make a difference. This is hard. For our kids that have multiple genes that are missing, this is a super big challenge because it's not just one gene that's a problem, it's many. So gene therapy is going to be, a major challenge. At some point, we may be able to do this. I was actually a part of a group that they tried to bring a whole bunch of people together to say, how could you attack this? And after really seriously thinking about it a lot, we just said, you know what, we don't have the technology to play this game yet. Like, we just can't do it yet. Maybe there'll be some breakthrough technology that will let us do it. A lot of you have probably heard about CRISPR. CRISPR is a technology that's being used to help change genes. It still has limitations, like significant ones. One of the difficulties is, again, when you have a big piece of DNA that's missing, it's not there to work with. Like, it's just gone. CRISPR and such often works with pieces of DNA that are just not quite right, and they just need to be tweaked. And even then, it has its drawbacks and problems. So sadly, we're going to get incremental better at doing some diseases, but 1P36 will be a unique challenge, very hard, just because of the large amounts of material that are missing. Are other kids in the same boat? Yes. Down syndrome has been looked at for such a long time, right? But when you have an entire extra chromosome, that's really hard. And so, anyway, this is a challenge. One of the things I always also would just say to be very cautious about, there honestly are a lot of people who want to take advantages of families who have money and are desperate. Be very wise. as far as going like out of the country to get help. I had one family who was really sad. They went to India, spent a lot of money to get to India, spent a lot of money to get some therapy in India, and they came back and they said, the person who did our therapy said this is the problem, and they gave me a microarray result. This was back when microarrays were first being used. And they were very excited about the fact that they had a microarray result that they could show me. When they showed me the result, it actually had been done wrong, like it had failed. There was no result on their microarray. It had been done incorrectly. And so all of the spots were made no sense, like there was no pattern. And I sat down and I said, this is what a microarray should look like. This is what your child's microarray looks like. This is a quality problem. They said, well, everything that they did was based on this. I said, all I can tell you is that there's nothing here. I felt bad because sometimes people say, well, there's no harm in having gone to India and done this, right? But this family is like $15,000 poorer having done this. I'm like, I could have bought a car. In fact, you could do a lot of things with $15,000, right? And so the cost is real. We want you to get therapies that work, but it takes time and you have to develop them and it has to be real. So just be very, very cautious. If your doctor's worried about you going somewhere and doing some therapy, it's probably because they really love you and they're saying, this is not a good place to go. And you're going to spend a lot of resources doing things that don't make any sense, because even if they have a good heart, they don't know what they're doing and it's not helpful. So that's my caution. I felt so bad for that family and I didn't know what to tell them. It's like, it's just not real. So I'm going to hang out, come find me, and again, do it. I'm going to leave sometime around after dinner-ish.
Okay.
So
You're here the rest of the day.
Yeah, I'm here the rest of the day. Yeah. Thanks.
Thank you so much, Dr. Scott.